fasted and the more starving state you get into, the more ketones you're going to make. Maybe I didn't talk about this. So where do we make it? The liver. I would say over 50%, maybe 60, 70% of the ketones are going to be made in the liver. So that's the primary place that they're made. It is made in the mitochondria, and that's because we're primarily using acetyl-CoA. Well, not primarily. We are using acetyl-CoA. So acetyl-CoA is going to be that starting molecule to make ketones. Acetyl-CoA is the starting molecule for making fatty acids. Acetyl-CoA is the starting molecule for making cholesterol. So acetyl-CoA is a really important molecule for all of these. There's a lot of acetyl-CoA in the mitochondria, and that is where we start making ketones. Sometimes we just call ketones ketones, and sometimes we call them ketone bodies. Either or, it's the same thing. There's 3 ketone bodies, or ketones. Acetoacetate, 3-hydroxybutyrate, and acetone. Acetone is a molecule that's not metabolized. It's a side product. When we make the other 2, we make acetone. The first 2 can go to tissues, be used for energy. In other words, we can convert ketones back to acetyl-CoA. So we can take acetoacetate, 3-hydroxybutyrate, and we can convert them back to acetyl-CoA. And what can we do with acetyl-CoA? We can put it through the TCA cycle and then through the electron transport chain, and we can make ATP. The other one, acetone, is a product that creates this fruity, it's kind of a nail polish smell that we can exhale. So a sign of that someone is in ketosis, which is high levels of ketones, will be the smell on their breath will be kind of this fruity, nail polish smell. But the other 2 can be used for ATP production, except for the liver and red blood cells. So the liver does not use ketones to metabolize them. It makes them, but it sends them out for the other tissues to use. The liver itself will use fatty acids for energy. It won't use acetyl-CoA. It won't use ketones. And red blood cells don't use ketones. Why? What do the red blood cells lack? Why do they not use it? No mitochondria. So there's no mitochondria in red blood cells, which means they have no use for ketones. They can't convert it back to acetyl-CoA. They don't have an electron transport chain, so they'll not use it either. So ketones do become elevated with fasting. If you're on a low-carb diet, so we've all heard of the ketogenic diets, the fad diet. And someone with uncontrolled diabetes will also have high level of ketones because their body doesn't know they have glucose in the blood. So they have high levels of glucose in the blood, but that glucose isn't getting into the cell. And if it doesn't get into the cell, then the body thinks it's starving to death. And so someone with uncontrolled diabetes will also have high levels of ketones. So again, it's just a fuel, and most of your tissues can use it. So your muscles are going to be the main primary takers of that ketones. So cardiac muscle, skeletal muscle, but also the brain. So the brain will still need glucose. It cannot survive just on ketones, but it will start switching to using more and more ketones if glucose is less available. If you're an endurance athlete, you tend to make more ketones during your activities, during your endurance exercise, and that makes sense because your body is it's using glucose, but it's switching over to using more fat. And these are endurance athletes. If you're a sprinter or someone, a power lifter, then you don't use ketones as much unless you're doing some sort of fasted starvation diet. But if you're an endurance athlete, you will tend to make more ketones in general. And again, you're really trying to spare glucose. You want to conserve glucose during long periods of fasting, starvation. So this slide, starting at the top, ketogenesis happens in the fasted/starvation state. So you're also going to be doing, so here's pyruvate. Pyruvate will no longer be used to make acetyl-CoA. That's what this bar means. It's blocking this. Instead of being used for acetyl-CoA, pyruvate instead is going to be turned into oxaloacetate for gluconeogenesis. We need to make new glucose with pyruvate if we have it. We don't want to turn it to acetyl-CoA because we already have lots of acetyl-CoA. When we're fasted, we do a lot of beta oxidation. So you are a better fat burner, or you're primarily going to be burning a lot more fat in the fasted state. And when you are breaking down fat or fatty acids, you're making a lot of acetyl-CoA. So if we go back to beta oxidation, that very last step, that thiolysis, using that thiolase enzyme, you break the fatty acid down into 2 carbons off, and those 2 carbons are acetyl-CoA. So whenever you do a lot of beta oxidation, you make a lot of acetyl-CoA. So acetyl-CoA is going to accumulate when you are in the fasted state and starvation state. Acetyl-CoA is going to accumulate, and what happens with that is acetyl-CoA will come together, acetyl-CoA increases, and they join together to form ketones. And the ketone bodies I have highlighted in red, again, you have acetone, acetoacetate, and 3-hydroxybutyrate. And don't have to memorize any enzymes, although I might have some listed here. I'm just going to mark them out so you don't have to know these enzymes. But acetyl-CoA plus acetyl-CoA, so 2 acetyl-CoAs will make acetyl-CoA. You add a 3rd acetyl-CoA to that, and you get 3-hydroxymethyl-gluteryl-CoA. It's a mouthful, I can't talk, but essentially that mouthful is HMG-CoA. I'm going to put it in blue here, HMG-CoA. You're going to see this guy again, and I want you to remember, when you see it, it's made out of 3 acetyl-CoAs. You put 3 acetyl-CoAs together, and you get HMG-CoA. HMG-CoA is going to be really important because that is one of the steps in making cholesterol. And it has a really important enzyme called HMG-CoA reductase. You're going to hear that again when you get to cholesterol synthesis. By the way, I talked to a student. I keep trying to talk to students that have taken the boards recently. One just came back to me. She said there were 40 questions on biochemistry, and a lot of them were on the vitamins, which is kind of cool. I like that they focus on that. And she said the other part, she got a lot on the regulated enzymes. So those were the 2 main things that she really remembered. It was a lot of vitamins and a lot of remembering which of the enzymes were regulated. Okay. So this is important not just for my exam, but for the board. So acetone, acetoacetate, and 3-hydroxybutyrate. These are the 3 ketone bodies. And how do we make ketone bodies? We make them from acetyl-CoA. Where do we make them? Primarily the liver. And what do we use them for? We use them to turn them back to acetyl-CoA so your body can make more ATP out of them. So ketone body synthesis in the liver. So the liver and red blood cells cannot use ketone bodies, but the rest of your tissues can. And here's your textbook picture. It's showing how we make them. Essentially, you have acetyl-CoAs coming together. Here's 2 acetyl-CoA. Here's a 3rd acetyl-CoA. So we have 3 acetyl-CoAs here to make this HMG-CoA. That's what this big mouthful is right here, HMG-CoA. And then essentially we make our first acetoacetate. Acetoacetate can then be used to make acetone on its way to making 3-hydroxybutyrate. So the gist of this is excess acetyl-CoA leads to the production of ketone bodies. There's 3 ketone bodies. Remember what they are. Remember also which one is not metabolically active, and that's acetone. So 3-hydroxy-3-methylglutaril-CoA. And sometimes you just get rid of the numbers and you just say hydroxymethylglutarate-CoA. I don't want you to be confused when you see these words, which are the same word, but they're written differently. So hydroxymethylglutarate is the same thing as hydroxymethylglutaril. They're the same molecule, HMG-CoA, which you'll see in cholesterol synthesis. Acetone is inactive. It's essentially exhaled in the breath, can be detected as a fruity, nail polish remover-like odor on the breath, and that will indicate a sign of ketosis, and ketosis just means high levels of ketones. You're getting to the point where it might start getting dangerous if you go into excessive levels of ketosis, because it can lead to ketoacidosis. So again, don't have to remember all these enzymes. They're here, but you can ignore the enzymes. Just memorize the 3 ketone bodies and essentially what they're made from, which is acetyl-CoA. So ketones, typically we don't have a lot in the blood, don't have to memorize the numbers, but it's usually maybe 1 mg per deciliter. Uncontrolled diabetes or starvation, you can get really high levels of ketones. So upwards of 35 milligrams per deciliter. Ultimately, most of your tissues can use it as fuel. So it's not a waste product. And I had ran into a chiropractor actually at the school, and he kept talking about toxins and accumulation of toxins when you're in the starvation. I'm like, what are you talking about with toxins? And he was like, ketones. I'm like, they're not toxins. Ketones are not toxins. The body can use them. They're very normal, very natural. It's only when you have really excessive levels that you end up with ketoacidosis. And at that time, sure, I'll call it a toxin. But before that, I won't call it a toxin. It's just a natural molecule that develops in the body. And if you get high levels of ketonemia, that's just high levels in the blood. Ketonuria, that's high levels in the urine. And if you have excessive levels, it will lead to acidosis because you will end up with high levels of hydrogen ions in the blood as well. So increased hydrogen ions in the blood. That's metabolic acidosis. Yeah.
You said blood will be, start to see 1 milligram per deciliter in our, American society, tend to basically never eat fast unless they're sleeping. So, what if someone can have fasted for, let's say, 24 or 48 hours? What would you consider level of target?
I have no idea. Yeah, I don't really know, and I'm sure someone has probably done like a time study looking at how quickly or how much ketones accumulate during the fasted moving into starvation. Someone has probably done a study like that, but I don't know how quickly, and it also is individual. Some people are much more metabolically active, they have a high metabolism versus other people, and they'll develop higher levels of ketones faster than someone who has a slower metabolism. But yeah, I usually don't know what those numbers would be. And because you're right, in America we eat all the time. So even this might be high, 1 mg per deciliter. We might not even reach that because I don't know about you, but I've been eating all day. Some days I don't eat, it's weird. But today I've been like snacking every 5 minutes, it seems like. So I doubt I have any ketones in my body right now. But it also depends, too, on how much of a fatty acid you're burner, or a fat burner versus a carb burner. There's a lot of variables that play a role in that question. So diabetic ketoacidosis is a real concern. Really dangerous. It can lead to death. Essentially, with someone with ketoacidosis, so they have low levels of insulin anyway, which means they're going to ultimately be their body is going to pretend or think that it's more fasted anyway. Even if it's got plenty of fuel, the body thinks it's fasted because there's high levels of glucagon. Glucagon leads to lipid breakdown or lipolysis, which means you're going to have a lot of free fatty acids, which means you're going to be making a lot of acetyl-CoA. And whenever you make a lot of acetyl-CoA, that excess acetyl-CoA is going to be turned into ketones, which means eventually you will become acidotic if it remains uncontrolled. And that can lead to coma and death. So it is fatal. Yes.
When I'm doing fasting, it start to warm up 12 hours.
And that makes sense because that's when your glycogen levels start to diminish. Well, I think like we need like a more control. He was asking like numbers as far as, I mean, I think that individually it's still, I don't know how the levels right, and especially with individuals that fast regularly versus individuals that maybe not fast on a regular basis. It will differ depending on the people. But an average, I don't really know what that looks like. But yeah, anytime we start becoming fasted, especially if we run out of glycogen, then that's glycogen, then your body will switch to being a fat burner. And I can feel when my body starts doing that. I can feel when my body is like switching fuel systems.
What does it feel like?
What's it feel like? Well, it's essentially if you can, like, it feels like you're, like I'm going, I don't know how to describe it. Like it's almost like the cold sweats. Like it's almost like my blood pressure is dropping. But it's not that. I'm switching to the next lecture. Oh no, I'm not, because I actually have questions at the end of this one.
Yes.
So, all right, the first question. I totally forgot to tell you guys about the hormones. CCK and secretin. So this is something I've been remembering, like I need to tell you guys about these hormones, not because I care. Well, I care that you know for the boards. I care that you know for the boards. Is it that important? Maybe. These are two really important hormones that could be on the boards. So CCK does two things. It tells the gallbladder, and actually I should see who watched my videos because it's actually in the videos. The gallbladder and pancreas, it causes them to contract. I don't know what that question was. Oh, so the answer to this question is, I'm going to write out the whole word. It's called cholecystokinin. Is that an I here? Cholecystokinin. Cholecystokinin. Okay, I'm not sure which one of these has the Y, as you can see here. But cholecystokinin is the enzyme we tend to just call CCK. And it tells the gallbladder and the pancreas to contract. It also will slow down the stomach emptying. Secretin, there's only one thing to remember that secretin does. And remember, CCK does everything else but this. Well, I shouldn't say everything. But secretin, it tells the pancreas to secrete something. Do you guys remember? To secrete, if you watched my video, you might remember. It's to secrete bicarbonate. HCO3 negative is bicarbonate. Why do you need to secrete bicarb into the intestinal tract? When you have food coming from the stomach, why does the pancreas need to secrete bicarb? Because of the acidity. Your stomach acid is so acidic that it can burn a hole through the intestinal tract. So what your pancreas does is secretes not hydrochloric acid. That's what's in the stomach. The stomach has hydrochloric acid, and so the pancreas will secrete bicarbonate in order to counteract the acid coming from the stomach. Bicarb is a base, it's alkaline, and that alkalinity will help neutralize the acid coming from the stomach. That's what secretin does. cholecystokinin does a couple of things. It tells the pancreas to contract, and it tells the pancreas to contract and secrete enzymes. To secrete enzymes. I gotta give you an apple pen. To secrete enzymes is why the CCK. I'll remind you guys of this before the exam. But the gallbladder secretes what? Why does we need the gallbladder to contract? What is it going to secrete? Bile. And then the pancreas needs to contract because it's going to secrete pancreatic enzymes. Remember pancreatic lipase, cholesterol esterase, phospholipase A2. So all of those are actually being secreted from the pancreas. So the pancreas has to contract. CCK is what tells the pancreas to contract. So secretin, the only thing it does is tell the pancreas to secrete bicarb. CCK will also tell the stomach to slow its emptying in order to slow digestion, because you don't want the food to get into the intestine from the stomach too fast. You want to slow that down. And so CCK will slow the stomach down too. So that's why I said CCK does most of the stuff. Secretin, its job is just to tell the pancreas to secrete bicarb. All right, so the first one you didn't really know, so I helped with that one. But you should know the other. So the answer to this, peptide hormone that's secreted in the intestine to trigger pancreatic bladder secretions for fat digestion, that would be CCK, exactly. What is the carrier for medium chain fatty acids in portal circulation? Exactly. What is the carrier for long chain fatty acids in circulation? It would be chylomicrons, exactly. But that would be for long chain. Hormone-sensitive lipase is covalently activated by, yeah, I'm going to read it too so you guys tell me. Okay, so hormone-sensitive lipase, does it come out when we're fasted or fed? Fasted. Which means you can get rid of insulin, so you don't have to read the ones out about insulin. So it means it's either B or D. And does glucagon activate a kinase or a phosphatase? A kinase. So yep, the answer is B. Glucagon via kinase, which phosphorylates the enzyme and activates hormone-sensitive lipase. So insulin activates a different lipase. It is called lipoprotein lipase, LPL. This one is activated by insulin. So insulin does activate lipase, but in this case it's lipoprotein lipase, which is going to break down chylomicrons. So it's in your fed state. Remember, chylomicrons are made when you're in the fed state. And to get the fatty acids from chylomicrons, insulin will activate lipoprotein lipase, and lipoprotein lipase will break down the lipids in chylomicrons to help bring those lipids into the adipocytes, into your fat cells. But hormone-sensitive lipase, that's glucagon. What is the end result of beta oxidation when oxidizing an odd chain fatty acid? Okay, good job. I heard it from some people, but not everyone yelled it. So, but yeah, propionyl-CoA. Where do we see malonyl-CoA? Is that from beta oxidation? What's it from? Fatty acid synthesis. We take acetyl-CoA and we carboxylate it and we make malonyl-CoA. That's the first step in fatty acid synthesis. All right, question number 4. Which hormone is considered to be an antagonist of lipolysis? I heard it. Insulin. So essentially, antagonist means against. Antagonist means against. It doesn't want to. It wants to prevent lipolysis. So the breakdown of lipids, you don't want to break down lipids when you're in the fed state. That happens in the fasted state. And so the antagonist would be insulin, because insulin comes out in the fed state, it's gonna keep you from breaking down lipids. Lipolysis is something that happens more in the fasted state. So all of these guys are going to stimulate lipolysis. Thyroxine is a metabolism hormone, glucagon, epinephrine is a stress hormone. What important molecule for the electron transport chain important molecules are produced after one round of beta oxidation using a saturated fatty acid? Is it, oh yeah, NADH, and what's the other one? FADH2. And how many do you make per round of beta oxidation? One each. So you make one NADH and one FADH2 after one round. So every round of beta oxidation, you make one of each. Would an unsaturated fatty acid produce more or less energy when undergoing beta oxidation? Less. So basically you don't make FADH2 everywhere that there's a double bond between carbons. You do not make an FADH2. So you make less energy when using an unsaturated fatty acid because you don't make an FADH2 everywhere that there is a double bond. Oh, that's another one. Which ketone body is not metabolically active? But the other two can be converted back to acetyl-CoA. That was it. All right, here's the review slide, so you can kind of go through and review questions. All right. Moving on to the next one. All right, so this one is phospho, and this one's not my favorite. It'll be fine though. PDF version. So this is the one, I want to really tell you guys what you need to know. There's stuff on the slides to try to help students understand, but let's keep this lecture simple, is what I want to do. Because these molecules are really complicated, their functions are really complicated. I don't think you need to know all the little details. But you do need to understand the membrane lipids. So chapter 17 is about membrane lipids. So we have different types of lipids. We have phospholipids, sphingolipids, glycolipids. Cholesterol is also in your lipid membranes, but we're not going to focus on this one today. So we're going to focus on phospholipids and glycolipids and just talk a little bit about their functions and their structure. And I just mean a little bit. Starting with this picture here, I guess. So this picture, these are your membrane lipids. So your cells making up your body, you need to understand kind of what those tissues look like, the molecules that make up these lipid membranes. These lipid membranes are phospholipids or glycolipids. And you can also have the two together. You can have a glycerophospholipid. So if you have a lipid with, so here's a lipid. If it's got a phosphate attached, that's a phospholipid. If it has a glycerol, which means, not a glycerol, this is a glycerol, but some of them will have like a sugar attached. So you'll have like a glucose or a galactose attached to it, then it's called a glycolipid. So a glycolipid, you have a sugar attached, so every time you hear glyco, think sugar, phospho, think phosphate. So one or the other, you either have a sugar attached to it or a phosphate, or sometimes both, and that will determine what that lipid's name is. So we have glycerophospholipids, which have just a glycerol backbone and a phosphate, and then we have something called sphingolipids. Sphingolipids can have a phosphate group attached to them or a sugar attached to them. So we're going to kind of, this is just an overview. You might be a little confused by the way I'm talking up here, but it'll make sense as we go through it. So here's a lipid membrane. Notice that you have these different lipids also hanging off of the lipid membrane. So we're going to talk a little bit about some of these lipids that are really important in the membrane because they have functions that are associated with the membrane itself. Yes.
Does the glycolipid have the sugar and the phosphate both?
No, a glycolipid will have a sugar. A phospholipid will have a phosphate. Having said that, you could have one with both. But for purposes of this class, we're going to separate them. A phospholipid, well, anytime you hear phospho, it's got a phosphate group attached to it. Anytime you hear glyco, it's got a sugar attached to it. So if I said phosphoglyco, then you would know it has both. But I won't do that in this lecture. I'll keep it separate. All right, so now we're going to focus. I'm going to tell you the details you really need to understand. Phospholipids are molecules that either bind to a glycerol or a sphingosine. So in other words, here is a glycerophospholipid. You're going to have a glycerol group, 3 carbons, 1, 2, 3, that's a glycerol. And it's going to be a diacylglycerol. So there's going to be 2 fatty acids attached and a phosphate group attached to this other carbon here. You're going to have a phosphate group. So a glycerophospholipid has a glycerol as a backbone and a phosphate group attached. So phosphate group. Versus a sphingolipid. So this guy does not have a glycerol backbone. It has it's kind of a similar structure. I'm going to show in the slide. That one I don't draw out very well, but the sphingophospholipid, instead of a glycerol, it has a sphingosine attached to it. So both are really important in cell membranes. And so we'll talk about the role in cell membranes and their lipid signaling molecules. So they also act as lipid signaling molecules. Keep in mind they're amphipathic too. Amphipathic means they have both a hydrophilic and a hydrophobic portion. Yes, and I'm going to show a picture in a second here. No. When I show the structure, because this is just introducing them, I'm going to show you the actual structure in a second here. But it doesn't have a sugar. But you do have glycerosphingolipids that do have sugars attached to them. I don't want to. It's so hard when I'm talking about something because you haven't seen the whole picture yet. So let's. We will get back to that though. Hopefully it'll make sense when we go through a few more slides and you see all the structures. And then I'll put them all together. So a glycerophospholipid, we're going to focus on this one first. So that slide was really just introducing them. I'm starting with the big picture, and I'm going to focus you guys into the details that you really need to know, starting with the glycerophospholipid. So glycerophospholipid has a diacylglycerol attached, a diacylglycerol and a phosphate group. So what's a diacylglycerol? Glycerol is a 3-carbon molecule. Diacyl, what's a triacyl? It means there's 3 fatty acids attached. So a diacyl means there's only 2 fatty acids attached. That's a diacylglycerol. And this last carbon here, because remember it's a phospholipid, is going to have a phosphate. So this carbon is going to have a phosphate group attached. So a glycerol backbone and the phosphate group. In other words, you have a diacylglycerol, a glycerol with 2 fatty acids, and that last carbon, that 3-carbon glycerol, has a phosphate group attached to it. That's its structure. And it is amphipathic, meaning you have this portion here, this head group is going to be hydrophilic, so it's going to like water. I'm just going to put water here instead of trying to write hydrophilic. It's hydrophilic though. And then it has this lipid portion down here that doesn't like water, so this is the phobic. It doesn't like water. This is the fatty acid tails, hydrophobic. So there, again, structure, I'll start down here. The structure is a glycerol backbone. It has two fatty acids, which is that diacylglyceride and a phosphate group. Plus, it typically has something else attached to it. Choline is kind of one of the most common ones, although we will talk about a little bit about inositol as well. But the primary structure is a glycerol backbone. It's got the two fatty acids, that's the diacylglyceride portion, and then a phosphate group. So again, they have a glycerol, that's why they're called glycerol phospholipids. They are the major class of phospholipids. They are the predominant lipid in membranes. Glycerol phospholipids is the one that I introduced you to last week or whenever it was, the first week of class. It's the most common type. It is made from phosphatidic acid. So phosphatidic acid is the simplest and it's the precursor of the other members of the group. So I have a picture up here of phosphatidic acid. And you see, it is the typical phospholipid. It's got the 3 carbons, glycerol group, 1, 2, 3. It's attached here to a phosphate group, and the other 2 carbons have a fatty acid tail attached to them. That's a phosphatidic acid. It's the simplest one, and then what we do with this phosphatidic acid is we add these other steps to it. Like if we added a choline to it, we can add an ethanolamine to it and a inositol to it. And so we can specialize these guys. But they're the most general, most common lipids in our membranes are these glycerophospholipids. This is just another name for it. So glycerophospholipid is the same thing as phosphoglyceride. So that same name, we could also just call this glycerophospholipid. It's the same thing, we just basically switched it up. We call it, but it's the same. Phosphoglyceride is a glycerophospholipid. And it's critical for maintaining cell membrane structure. That's why it's the most common one. But it does facilitate signal transduction. So when you have a cell membrane, you need to send information, you need information to go into the cell, and so these phospholipids are going to help with that cell signaling. So they're going to help with signal transduction. And they do other things, so cell communication, lipid metabolism, there's a variety of functions, but their main function is the cell membrane structure, and we're going to talk a little bit about cell signaling. Here's some examples. So phosphatidylcholine is phosphatidic acid is the simplest. It's the precursor. Here's cardiolipin. Here's phosphatoethanolamine. Here's platelet activating factor (PAF). So these are all just examples. And as far as I'm concerned, you can ignore them. You don't have to study them. But these are all examples. If you add stuff to the phosphoglyceride, you will specialize these phosphoglycerides into some specific type of function. And I could go on for days about all the different functions that these different ones have. These are the 2 I want you to know. These are the 2 most important phosphoglycerides in humans. Phosphatidylcholine and phosphatidylinositol. Phosphatidylcholine is the predominant lipid membrane lipid. It's the most abundant phospholipid in the human body. So typically when anyone talks about phospholipids, they're typically talking about phosphatidylcholine. It maintains the integrity, the fluidity of the cell membrane. It's the major component of lipoproteins as well. And it is derived from choline. So that's the specialized product that's attached to it. So you still have the diacylglyceride, the glycerol backbone, the 2 fatty acids, and you have the phosphate group, and on this phosphate is actually a choline. So you have a choline attached to that phosphate too. That's phosphatidylcholine. predominant lipid in your cell membranes. This other one has a lot of different functions. It's called phosphatidylinositol. It is a really important.
Your screen's broken.
Oh, you guys didn't even see it after I was drawing on that. That's cool.
See, it's like the old ones, I'm just saying, it's 3:50.
Yeah, we need to take a break. It's definitely time for a break. All right. Wow.
It's a process of it. It goes into a process. Yeah. So, but actually in our bodies, when we don't eat that's what but it's okay to do it. The problem where the problem is if you still like sleep, where ketones start to work and starts to this happens is
So this is C6, C7, T1. C7, T1. This one, C7. Wait, this one? This one? I'll tell you right now. That's C7. Remember that the one after foods that like stays there regardless could be C7 or T. Yeah. Like literally C7 and T1 are literally like they're like on each other like this. Just like on top of this. But the reason why I know is because C6 for sure is the one that goes away when you go on that. So you can always find C6.
Hello there.
Yeah, I'll probably be at the library this weekend at some certain points.
Yes.
So I can work on it then. I was thinking maybe I could today, but I was like the dog specifically. I left at 12, so I'm like I can't leave it alone for like 7+ hours.
I hear that.
But I definitely will be in this weekend, and maybe I could do like Friday and Saturday, or Friday
Yes.
and some Thursday as well. But
Whatever works as long as we get it done. It's good.
we do need to start grinding it out. Because when's it due? Week
7, yes. It's already preparing for midterms. It feels like we just started. I have about as much information in my head as
It's gonna be another cramming session.
absolutely.
We'll get it done now.
Yeah, I believe you.
Which one are you least looking forward to right now?
As far as believe it or not, Neurophys shouldn't be that bad. According to the later quarter people, they say that his test is consistent with his study guide.
I didn't know he had a study guide.
He should have a study guide. I wish he posted today, but
Pace was saying, I think Visceral Fizz, that he changed a lot of his exam questions. You heard about that?
no, I did not. Tell me.
She said that he changed, like it used to be a lot of his would be on the, like, quizzes we did, and knowledge checks, but apparently he changed it, so like now it's different from the knowledge checks and stuff like that. From what she said at least. I'm still going to study the knowledge checks.
Oh yeah.
But like, I don't know how different they are.
It's gonna be rough. It's gonna be rough.
We're just gonna once we graduate, I'm just gonna get like life tattooed across my chest.
Exactly. I survived like you.
Yeah. Maybe it could be like one of those like you remember that Subway thing where if you get like a 12-inch tattoo, you get free Subway for life.
Exactly.
Maybe if you get like a whole back piece, they'll pay for your student debt.
Better yet, I just tattoo my hands black. That way I can do the the sign out front or bronze. I don't want to do black hand.
Bronze hands would be hard to pull off. Just always
You're not
have
supposed to pull them off. That's why they're attached to the end.
have self-tanner on. It would look crazy with your Eldritch theme.
Absolutely.
With your Eldritch theme office.
Could you imagine that? It's like my hands are coated in the.
People would be like, he definitely practices some black magic.
Exactly. Never mind all the runes he has going up his arm. We're not going to worry about that.
Yeah, that's how I was confusing.
Yeah, definitely this weekend and then
Yes.
sometime next week. 'Cause next weekend as well, I'm watching the dog, but next week during the actual weekdays,
Gotcha.
I am not. So.
We'll say for this weekend, I'll probably have enough flashcards down to make us happy about having flashcards, so we can get that done because we need to do like actual studying in addition to class stuff and clubs and things like that. So
That's what, man. It's so fast. It's like hard to get clubs in because of how fast
yeah,
paced it is.
well, yeah, it's bad.
Are you going to that one club again Thursday?
I want to see about Torque Club, see how that is. Which one was that? Was that we went to Applied Kinesiology? I may go to that again. Just to I don't plan on joining. I plan on going like occasionally to see what happens. There was that other applied neurology one that's interesting. I don't know if you've been. Yeah, have you been going to your weekly sessions or whatever they are? The adjustments?
Oh, with my student intern?
Yeah.
I've been going. Last week she was all booked up though, so I wasn't able to. But I do have a student intern. who I see
Gotcha.
every week.
Okay, so do they ever do the like make you trace your follow your thumb?
I think only like one time she checked my eyes to make sure it followed, but that was during the assessments they do.
Gotcha.
So I think
My intern is consistently doing that before and after to see how well my nerves are tracing and something like that. That's what
maybe
applied
maybe it's for different stuff.
No, it's for different stuff, but it's like apparently before the adjustments, my eyes kind of like jump from like spot to spot, and afterwards it was much more smooth. So
Afterwards, your pupils were like super tight.
exactly, you know how it is.
We just gotta make it to clinic. Someone told me clinics where a lot of people fail actually just not getting enough hours apparently.
That doesn't surprise me, to be completely honest. I've seen courses and things like that have required number of hours and things like that, required number of X, Y, and Z. So what I'm really hearing is we'll just have to go and do it for each other. Like, hey, I need this
We
coming.
will have to probably work with each other quite a few times.
Yes.
So I'm definitely once we get there, me and you need to team up. But someone did say, I think it was Victoria. She said that she talked to the guidance counselors, and they said usually if students can pass, I think second quarter,
We should be good,
then they're good. Because after that point, like it doesn't, not necessarily gets easier.
but it's consistent. Or
But
it doesn't get any harder.
Yeah, at that point, that's like she said, that's a pretty consistent showing of like people who are willing to put in the work.
All right, but I know what I need to do.
So we just have to pass
We have to do this. Yeah.
which we'll see. I don't know my least favorite classes right now, but
None of these are very good. None of these are very good.
it might be micro. I have not paid attention to micro just because like
There's no paying attention to micro.
no one is.
No.
No one's showing up, and then those who do have headphones on. Me included.
I feel so bad for her because whenever I'm the only person who doesn't wear headphones. So whenever she asks, well, do you know this question? She looks at me because I'm looking at her.
I think Sarah might be the only person paying attention to class because she like answers the questions too.
Yes.
I don't like it's just too much. If I tried to do microbiology like I did the other classes, I just
Oh, there's way too much there. The PAL is good for microbiome. He actually he took. Okay, so Dr. Liu has been doing this for like 30 years. So all the information she has in her head is what she's trying to put on paper. She's not putting our level on paper.
Yeah.
So the PAL session guy is able to put it on a level and dumb it down. Now I don't want to say dumb it down because it is still the same thing, but it's
But I
much
understand.
less
Yeah, like
involved. It's
that's what you can just tell. Like she is like throwing a textbook at you, kind of like that's the prompt of hers.
yes.
But you can tell she knows the stuff.
Oh yeah.
With the neurophysiology, apparently he's a neurophysicist.
I guess.
But it feels like he doesn't know the material as much. Like,
Really, he just does not seem confident in his ability to portray it.
maybe he's just not good at explaining things.
Occasionally, he'll have a moment of clarity, and I'll be able to gain something from him, but it's not very often.
I know that was brutal earlier.
I was in the back of the class. I was not.
I was wondering. I was like, I didn't see the first half, and so I was like,
I went to Walmart to get this big hunk and a text or notebook here. I'm completely revising my notes, the way I take my notes, because the way I'm doing it right now is not working.
yeah, I think I might need to do something like that soon.
I am debating whether I want to return the iPad.
You want to
I'm
what?
debating whether I want to return the iPad or not.
Yeah, that's fair. I mean, it's not for everyone, but
It's probably not for me. I'm spending too much time worrying about how to format my notes and then actually writing notes.
and hey, if you buy it back, it'll be $200 off again.
I don't know. I feel like I'm just running around in circles.
Yeah, it is. It's definitely. It's not for everyone.
I'll have to ask.
It's just more convenient for me personally.
Yeah, I'll have to ask Best Buy what their return policy is and keep it for until then. I still have the boxes in my room. And I'm glad I got it, but I dug where'd it go? It's in one of these bags. The case that this came with,
Yeah.
I threw away the box. I was like, yeah, you know what? I'm gonna keep this. I was like, nope, I don't know what I'm doing with this.
Maybe just like either give it away to someone or see if you can sell it to someone.
Sure, why not? I'll see it. It's really not that. It's not that difficult. It's just too many little things getting in the way to make it.
I remember the jump from Android, Samsung to iPhone was weird. And so it's going to be weird definitely for you to iPad.
So, do you think it would be any easier for me to use a Samsung tablet?
I don't know.
Let's kick off some stuff that I have in here, I think. So luckily it seems to be working. It's not my iPad, it does appear to be the Screen Beam. So Screen Beam just doesn't seem to want to cooperate. So
Draw
hopefully
something.
it'll keep cooperating.
Draw something.
Draw what?
Something. There we go. It worked. Perfect. Real time. Oh, that's.
Let's see what's going on over here, dog.
There's nothing going on here. Right there.
That's
Well, it makes the world go round.
Feel that sound?
Alright
Which
guys,
that's
so phosphatidylcholine.
dang.
real tense.
Most
Oh,
important
so
one,
tense.
It's always been like that.
phosphatidylcholine.
Always.
Essentially guys, it has
It has never
three
not been like that.
functions. This is all I need you guys to
Please.
know are these three functions for phosphatidylcholine. Well, phosphatidylcholine, its function is membrane integrity, fluidity of cell membranes. It is the most common component of your membranes, your cell membranes. Phosphatidylinositol is a little more complex. It has three functions. One, it's really important in cell signaling. Okay, I just blocked that. It's really important in cell signaling. So it serves as a precursor for inositol phosphates, which are really important messengers in the cell membrane. So basically, if this heart is a cell, that essentially then inositol phosphate, which is IP3, so if something binds to the cell, it can trigger a cascade of events that will, the inositol phosphate is part of that membrane, IP3, and can send that message into the cell. So IP3, really important second messenger in cells, help provide the message coming from the outside to the inside of the cell. Because it's sitting on the membrane, it has access to the information, and it can provide that information about what's going on outside the cell into the cell. And the other one is diacylglycerol, which is just a common fat, but it's also a it can act as a messenger. So these are just signaling messengers that are part of the function of phosphatidylinositols. The other function of phosphatidylinositols is as a reservoir for arachidonic acid. Remember the importance of arachidonic acid. It's going to be really important for making our eicosanoids. That's an E. Eicosanoids are these hormone-like molecules we're going to talk about more in a second here. So one of the fatty acids, because remember we're talking about phosphate groups, so we have two fatty acids here. One of these fatty acids is going to be arachidonic acid. So arachidonic acid is going to be one of these fatty acids, which is going to be really important when the cell needs to synthesize eicosanoids. It can just go in and break down one of these phosphatidylinositols and take it to arachidonic acid. So a reservoir for arachidonic acid is another important function. That's function 2. So first function, cell signaling, it makes inositol phosphate. Really important messenger for signaling messenger. Second function is a reservoir for arachidonic acid. Third function is protein anchoring. So essentially what that means is phosphatidylinositol will actually help attach stuff to membranes. So phosphatidylinositol, remember it's sitting in this membrane here. This, let's say this blue here is the phosphatidylinositol. It can actually act as an anchor. In fact, it does hold various enzymes to the cell membrane. For example, it holds, let me write it black here, lipoprotein lipase. We've seen that before. It breaks down triglycerides in lipoproteins, chylomicrons. And so lipoprotein lipase is not inside cells. It's outside of cells. It's on actually the cell membranes of your blood vessel walls. And it can actually hold on to lipoprotein lipase so that when chylomicrons are swimming by, it can attack, the lipoprotein lipase can attack those chylomicrons that are swimming by in your blood and pull out those triglycerides. So the function here is an anchor. It anchors lipoprotein lipase and other proteins and enzymes to the wall of the blood vessels or wherever it's at in order to keep them stationary there so they can do their function, whatever that is. So protein anchoring. So function 1 is cell signaling. It serves as a precursor for inositol phosphates, IP3. Function 2, it's a reservoir. One of the fatty acids in there is arachidonic acid. We need that for eicosanoids. Function 3, it helps anchor various proteins and enzymes wherever it needs to be. So for example, lipoprotein lipase can be anchored in the blood vessel walls and attack the chylomicrons as they're swimming by. And that's the function of phosphatidylinositol, which is a type of phosphoglyceride. So remember, these are just 2 types of phosphoglycerides, and these are the only 2 that I care that you remember. There's a lot of others. I just chose 2 of the more important ones. These pictures are just from your textbook. Phosphatidylinositol, this is just showing how one of the fatty acids is arachidonic acid, and it can actually cleave that guy off, and that arachidonic acid can be used for eicosanoid synthesis. So it's a reservoir for arachidonic acid. This picture, I made it really small because I don't want you to study this. I just want you to know that phosphatidylinositol makes really important messengers, signaling messengers, especially IP3. And this is showing you what those second messengers do. They help activate enzymes. They help release calcium that helps them move various other compounds in the cell. So they're just really important for helping integrate the cellular communications so that the cell is communicating and operating effectively. So communication is key. And phosphatidylinositol helps produce those communicators like IP3. And this one, this is another one. Again, you don't have to study all this, but this is just showing protein anchoring. So see, phosphatidylinositol actually sits in the cell membrane here, and it can anchor stuff to the cell wall. So it's going to be able to anchor a protein. So here's its anchorage. It's anchoring, in this case, this protein, for example, lipoprotein lipase could be anchored to the cell wall here thanks to phosphatidylinositol. Plus it's just fun to say, phosphatidylinositol. Phosphatidylinositol. Everyone together now. Phosphatidylmethol.
Supercalifragilisticexpialidocious.
It's not easy, is it? My job is hard, people. Okay. So those were the glycerophospholipids. So again, two glycerophospholipids you need to remember are phosphatidylcholine and phosphatidylmethol. I don't know how to do an acronym for that one. But those are the two glycerophospholipids you need to know. And just generally understand their structure is a glycerol backbone, two fatty acids, and a phosphate groups on that last carbon there. Sphingolipids. This is going to be simple. Sphingolipids have a sphingosine and a phosphate group. So sphingolipids, sphingosphospholipids have a phosphate group. Not all sphingolipids have a phosphate group, but sphingosphospholipids have a phosphate group and a sphingosine.
Can you say that again?
Sphingosphospholipids. Sphingosphospholipids. Yeah, no, they're actually fun to say. Like, there's nothing else they're fun to say. All right, so the structure. I don't like to draw a sphingosine because I will always get it wrong, but I do know that sphingosine has a nitrogen group, an alcohol group, and two fatty acids. Or sorry, one fatty acid. So they have a nitrogen and hydroxyl group and a fatty acid. Honestly, I don't care if you remember that, but you should remember sphingosine. So sphingolipids have a sphingosine attached to them. And the sphingosine is in green here. The green part is still working. You have the hydroxyl group, the nitrogen group, and a fatty acid. And then if you throw another fatty acid on this thing, you get a ceramide. Yeah, it's cool. That's great. Okay, so sphingolipids are a diverse group of lipids with a sphingosine backbone. So sphingosines, again, don't have to remember all this detail, but it is just it's a fatty acid, a nitrogen, and an alcohol coming together. That's a sphingosine. And a ceramide contains a sphingosine plus just another fatty acid. So here's a ceramide, and here's the other fatty acid that's attached to it. So that's why I went from a sphingosine to a ceramide. You basically just added an extra fatty acid, and now it's a ceramide. So you still have two fatty acids, and this one has a choline. It doesn't have to, but see the phosphate group is here. So there is a phosphate group if it's a sphingophospholipid. But the key difference between a sphingolipid and a regular glycerol lipid or a sphingolipid has a sphingosine attached. A glycerol lipid has a glycerol group as the backbone. So the sphingosine makes up the backbone in sphingolipids. So there's no glycerol, is what I'm trying to say. There's no glycerol group if you have a sphingolipid. Instead of a glycerol, you have a sphingosine. Ceramides actually have a lot of really important functions. So you have spheramides and sphingomyelin. I'm trying to remember what you guys know. But sphingomyelin is a really important component of nervous system, specifically the myelin sheath. The myelin sheath of the nervous system. It has a lot of sphingomyelin. And a couple others we'll talk about are gangliosides and all right, we'll get to it though. But ceramides have a lot of really important functions, especially in the nervous system, helping with mostly signaling communication in the nervous system. So here's the structures and functions. So these are the type of sphingolipids you need to know. One is a sphingophospholipid. The other is a glycosphingolipid. So a sphingophospholipid, how it differs from a glycophospholipid, or sphingophospholipid have a phosphate group, and glycosphingolipids have a sugar instead of a phosphate. Yes, you cannot have a sphingolipid without a ceramide. A sphingosine is the main backbone. You add a fatty acid to that, you get a ceramide. And a ceramide is the major component of sphingolipids. I mean, sphingosines are as well, but technically, sphingosines are always, well, I shouldn't say always, but most of the molecules that have a sphingosine are actually eventually produced into a ceramide. You throw an extra fatty acid on it and you get a ceramide. So both sphingospholipids and glycosphingolipids contain a sphingosine, which is made into a ceramide. So technically, the sphingosine is considered the backbone, but a ceramide is a component that's added to that sphingosine, or a fatty acid added to the sphingosine, you get a ceramide.
It's not a precursor, it's.
Well, sphingosine would be a precursor in the production of ceramides, you could say. Like you just throw an extra fatty acid onto it. I mean, you could say it that way. But the sphingospholipids have a phosphate group, and they are the structural component of a variety of cell membranes, especially the myelin sheath of neurons. That is predominantly where you're going to find sphingospholipids. Sphingospholipids primarily are in the myelin sheath of your nervous system, which is important for the structural integrity of those nerves. Yeah.
So what is the choline coming to this diagram? Is that part of the sphingospholipids with the ceramide?
So, actually, I was just looking, is the choline is not always there. So, yeah, I want to say that the choline doesn't always have to be there. It could be something else there. Well, it'll still make it a sphingolipid, and it'll still make it a ceramide. It's just not specific to the ceramide or the sphingolipid.
And then if you go back to slides, is it the fatty acids that are the basic building blocks of this sphingolipid, or?
So the sphingosine is considered the backbone, and the sphingosine is all of this. All that's in green, I should say. All that's in green is the sphingolipid, and the way textbooks, it's basically, you do have, it looks like a glycerol, but technically it's not, because you have a nitrogen and hydroxyl, and so it's different from a regular glycerol. And technically that's the backbone. So instead of having a straight glycerol, you have a sphingosine, which has a hydroxyl group and a nitrogen group, this fatty acid, and technically that sphingosine is then altered even more by putting another fatty acid on it, and now it's called a ceramide. And this choline isn't not necessarily have to be there, but it is a common addition to a lot of these phospholipids. It's usually attached to that phosphate group, but it doesn't have to be there. It could be a serine or some other molecule attached to the phosphate.
And that phosphate will only be there if it's under phosphate attack?
Nope, that phosphate group, it just depends on what the cell is making. A sphingophospholipid has a phosphate group attached. A glycerosphingolipid will not have a phosphate group. Instead of this phosphate group, it's going to be a sugar. You're going to have a glucose attached instead of that phosphate. Yep. These are good. This is helping focus you guys. So the sphingophospholipid will have a phosphate group. There we go. Will have a phosphate group, whereas a glycosphingolipid will have a sugar in place of that phosphate group. And we just haven't seen a picture of that one yet. But instead of a phosphate, you'll have instead of a phosphate, you'll have a sugar. Like usually do I have it in there? A glucose or a galactose. Another main difference is sphingofospholipids and glycerosphingolipids is that sphingofospholipids are more in about the structural integrity of the molecule, of the membrane, whereas glycerosphingolipids, glycosphingolipids, I keep doing that, don't I? Glycosphingolipids, these guys are more important for cell signaling. Cell signaling are the glycosphingolipids. The sphingofospholipids are more important for more the conduction of your nerves and the integrity of your nerve membranes. So both sphingolipids, sphingomyelin and glycosphingolipids are vital for cell membrane structure, but sphingomyelin is primarily involved in membrane stability and nerve function. Myelinated, making the myelin sheath of your axons, whereas glycosphingolipids are more involved in cell signaling, recognition, interaction. So those are just functional difference between the sphingofospholipids, which are mostly made up of sphingomyelins, and the glycosphingolipids. I wonder if this is hard talking about this. All right, sphingolipids. So sphingomyelins again, so this is just kind of review. They're structural. They're similar to phosphatidylcholine. They're really about structure and creating the membrane. The key feature does have a phosphorylcholine. That's a major part of sphingomyelin. So it's really common to have a choline here attached to that phosphate group. I don't care if you remember the choline for this one, but you do need to know that sphingomyelin is a major product of sphingofospholipids. So sphingofospholipids, a major example of them are sphingomyelin. And these are widely dispersed in your nervous system. You have a lot of sphingomyelin. I want to get in a little bit into physiology, but I don't want to throw us off biochemistry, so I'm going to not do that. It wraps around your axons and helps with nerve conduction. So if you have myelin sheath, will help the conduction of your nervous impulses be faster and stronger. So myelin sheath, the same thing. Yeah, the sphingomyelin is the myelin sheath. Yeah. Yeah. Where I don't want to, I can get into that, but we don't have time, unfortunately. Although, even though I might think it helps a little bit. This is the picture, so it's just a really big picture. Again, sphingosine. It looks like a glycerol. We do have that 3 carbon there, but technically the difference is that we have that hydroxyl group and nitrogen. And we always add an extra fatty acid and make it into a ceramide. So, but technically the backbone of a sphingolipid is a sphingosine. The backbone of glycerophospholipid is a glycerol group. So Niemann-Pick disease is I don't want to say it's common, but it is caused by the being unable to degrade sphingomyelin. So as sphingomyelin accumulates, especially in tissues like the liver tissue, then you'll end up with Niemann-Pick disease. And this can All right, I'm just confused type A and type B, so now I am not comfortable with this slide.
We could break early.
So this won't be on the exam, because I've already forgotten. Type B, crap, okay, never mind, all right, apparently. But if you can lack the enzyme that degrades sphingomyelin and it accumulates, and depending on what tissue it accumulates in, it can cause disease, and it can be fatal. It actually is a neurological issue, but I've forgotten which type is A and B, so, but I could say if you don't properly properly degrade sphingomyelin, you'll end up with Niemann-Pick disease if you lack the enzyme that will degrade and break down the sphingomyelin. But I won't ask that on the exam. But here's the accumulation of the sphingomyelin. So anytime you see white in your body, that indicates fat accumulation. And what are lipids? Lipids are fat. And so essentially this white is indicating you have a lot of deposits in the spleen, and the lipid is primarily where you get deposits of these. And that's because that's normally where you break these guys down and degrade them in order to remove them from the body. So the liver and the spleen will end up with large deposits of sphingomyelin, which means those it becomes non-functional anymore. When you have too much accumulation, then it's hard to use those organs where it's accumulated in. All right. So that was the sphingophospholipid. Now we're going to talk about glycosphingolipids. Glycosphingolipids essentially have, here's the ceramide. So here is the, this guy here is the sphingosine. And then this is where we attach another fatty acid. So we have another ceramide, but in this case, instead of a phosphate, so there's no phosphate here, instead of a phosphate, you have a sugar. And that's galactose in this case, but it could be glucose. So they have a sphingosine backbone that forms into a ceramide and a fatty acid, so a ceramide, but instead of a phosphate group, they have one or more sugar molecules attached. These guys are more involved in cell signaling. So the sphingophospholipid, that's more structural sphingomyelin. These guys, the glycosphingolipids, the ones with the sugar molecules attached, when sugar molecules are attached, they tend to be involved in cell signaling and cell-to-cell recognition. And so the type of carbs, the complexity of the carbohydrates that are attached to it, determine the function of that glycosphingolipid. There's two types. You don't have to know the difference, but you should recognize that cerebrosides and gangliosides are two types of glycosphingolipids. Just like sphingomyelin is a type of a sphingophospholipid, you should know that cerebrosides and gangliosides are a type of glycosphingolipid. And these guys are not so involved in integrity, like the sphingomyelin. They're more involved in cell signaling and cell-to-cell recognition, like communication. They're involved in communication. So, again, you don't have to remember the differences, but they're cerebrosides and gangliosides. They're highly predominant in nervous tissue, just like sphingomyelin. So the sphingolipids are highly abundant in nervous tissue. It doesn't mean they're not in other tissues, but they're highly abundant in nervous tissue. And cerebrosides, the main difference between them are primarily related to their location. So gangliosides are more in the ganglion cells. The ganglion cells are the primary cells, the cell bodies of your central nervous system, versus cerebrosides are more found in the myelin sheath. So it's highly associated with the sphingomyelin component of the myelin sheath. Hopefully that didn't confuse you. And here's just another picture. Here you have, this is a cerebroside. This one's another glycosphingolipid. So here's the glycosphingolipid, and here's the sugar group attached to it. So again, the main difference between a glycosphingolipid and a phosphosphingolipid is one has a sugar attached, the other one has a phosphate attached. All right. Eicosanoids. Any questions about sphingolipids? I know you guys are just kind of to review this. So again, for these, keep it simple. Just kind of have a general understanding of the structure and a general understanding of the function. Okay, eicosanoids. I hate that I teach biochemistry at the end of the day. I would rather start the day with this. After you guys have just had your morning coffee, let's just do some biochemistry then.
Yeah.
Yeah, I don't know if I can convince them to change everyone's schedules around them. This is review though. This is a little bit of review, and again, I'm gonna keep it simple even though there's a lot of words on here. Eicosanoids, you know what those are already. They are prostaglandins, thromboxanes, and leukotrienes. And they are made from omega-3 and omega-6. You make omega-3 and omega-6, these guys come, omega-3, omega-6, these come from your diet. Remember, eicosanoid, eicosa means 20, that's where it gets its name, eicosa means 20. These are hormone-like substances. They do a lot of stuff, inflammatory responses, but they also do a lot of platelet movement, so they make platelets, blood clotting, they work on muscle contractions, they can change blood vessel diameters, they have a lot of function related to smooth muscles, intestine, uterus, so they have a lot of functions, but for the purposes of this class, we're going to focus on their function in inflammation. So they have a lot of hypersensitivity reactions, and we're going to focus on them in relation to inflammation. And they are hormones, they act like hormones, but they're not hormones because they don't travel very far. They only travel very short distances. They really are working on the tissues that are immediately in their vicinity, and they don't last very long. Most hormones can travel far distances, and they last for hours and days sometimes. These have a very short half-life, they don't last very long, and they really don't travel very far. That's why I say they're hormone-like, because they're not technically hormones. Eicosanoids are not technically hormones, because they're much more local, and they don't last very long. And they're not stored, again, they have an extremely short half-life, they're rapidly metabolized and made inactive pretty quickly. And here's their structure over here, but don't worry so much about memorizing their structure, although I will show you how big they are. They have lots of carbons in them, so this guy, they should be 20 carbons. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. 20 carbons in that. So these are 20 carbon molecules, all made from 2 essential fatty acids. We have alpha-linolenic, which is omega-3, and here's linoleic, which is omega-6. And yes, you should know the difference. Linolenic is omega-3, linoleic is omega-6. And they are unsaturated. So omega-3 actually has 3 sites of unsaturation. Omega-6 has 2 sites of unsaturation. And the reason we can't make them is because we do not have the desaturases, these enzymes, that make the double bonds in those exact spots. So we cannot make them, we have to get them from our diet. We do not have enzymes that can make those double bonds in those exact spots. We do have desaturases, but not the ones that can make this. So prostaglandins, thromboxanes, and leukotrienes are the eicosanoids. They are 20 carbon molecules, and they're formed by incorporating oxygen into the fatty acid chain. And so that's why they're going to be made by something called COX enzymes. There's one other one, it's called a LOX enzyme. So this O is representing the oxygen that's needed to make them. So oxygen is going to be put into them by these COX and LOX enzymes. And I'll show you what that looks like. So here's the COX enzymes, so technically called cyclooxygenase. There's a couple of them, COX-1 and COX-2. You don't have to remember which one does which, but they both are involved in platelet aggregation, which is blood clotting. So that's forming a blood clot. And the other one is involved in inflammation. So both of them are part of the healing of the body. They're making the body better. If it's injured, inflamed, sick, if there's damage to the tissues, that's when you're going to be making these eicosanoids. COX-1 and 2 make 2 of them. Thromboxanes and prostaglandins are made by COX enzymes. These activate the immune system, cause vasoconstriction, inflammation, and platelet aggregation, generally speaking. So they clot your blood and make you inflamed. They also close your, the other one closes your bronchioles. So lipoxygenases are made by LOX enzymes. That must be in the next slide. And these are the ones that are part of the allergic response, also do inflammation. They close up your air pathways. And COX-1 and COX-2, I'm going to go back to those for a second. COX-1 and COX-2 are inhibited by aspirin and ibuprofen. Aspirin and ibuprofen, the reason they work is they block these enzymes. If you block, so ibuprofen, if you take that, it blocks these enzymes, which means that you are not going to, it's going to decrease inflammation. If you take aspirin, remember, I'm asking you guys to remember when, you probably don't remember, back in the 90s, they started recommending that the older people take these little baby aspirins, and that's because aspirin will block these enzymes, and it acts as a blood thinner, because these COX enzymes result in platelet aggregation. And so some people still take a baby aspirin to thin their blood if they have high risk of heart disease, if their vascular system tends to be more clogged, then they will take a baby aspirin to help thin the blood. Yeah.
So it's inhibited by aspirin and what?
Ibuprofen.
So NSAIDs are called blockers.
Non-steroidal anti-inflammatory drugs. Yeah, NSAIDs. NSAIDs block the yeah. Exactly.
That's actually really interesting. They say that
You think about it, yeah. Great. I love it. All right, so, linolenic is omega-3. So linolenic actually doesn't make arachidonic acid. It makes icosapentaenoic acid and docosahexapentaenoic acid, DHA.
Start over. What was that?
No, I'm not doing that again. I'm not doing the DHA again. DHA and EPA. But these are also made by cyclooxygenases and lipoxygenases make these guys. I'm going to say the full name now. I'm just kidding. So the COX and LOX will make the prostaglandins, leukotrienes, and thromboxanes. The difference between omega-3 and omega-6 is that omega-6s tend to be inflammatory. So omega-6 makes arachidonic acid versus omega-3 makes EPA and DHA. I do have it written out somewhere. Docosahexaenoic acid. Yeah. DHA and EPA, these guys tend to be anti-inflammatory, inhibit platelets. So whereas omega-6 activates omega-6s are used to make arachidonic acid. Arachidonic acid makes inflammatory, platelet aggregating prostaglandins and thromboxanes. Omega-3, it makes EPA and DHA. These tend to be anti-inflammatory, and they thin the blood instead of causing blood clotting. And so it has opposite effects. That's why it's really important to have a balance of these in the diet. If you eat excessive amounts of omega-6s, which makes arachidonic acid, then your body will be out of balance. It's going to be more inflammatory. You want that sometimes in order for your body to heal properly. But you should also have omega-3s in your diet to balance that out, because you also need anti-inflammatory sometimes. You also need your blood to be a little thinner and not clot excessively. And so it's really about a balance of getting omega-6s and omega-3s. It's recommended you have 3 times more omega-6s in your diet than omega-3s. I don't really know where they did that research at, but I will say that you need a balance of both in the diet. Both are important. And that's what this slide is kind of breaking down. Omega-3s make arachidonic acid. These tend to be inflammatory, cause vasoconstriction, contract your respiratory system, versus omega-3s, icosapentaenoic acid and docosahexapentaenoic acid. These guys tend to decrease blood clotting, are anti-inflammatory, and these guys need to be made just as much as the inflammatory ones. So both of these enzymes are using the same, both of these pathways use the same enzymes, COX and LOX. And so if you take aspirin and ibuprofen, it'll block both these pathways. But because the omega-6 pathway, arachidonic acid, is the most active one in the body, that's the one we see the effects of when you take ibuprofen and aspirin. They're anti-inflammatory, they help decrease inflammation because they're blocking this pathway. So, I shouldn't have erased that. So this is where your NSAIDs come in. Aspirin and ibuprofen and aspirin are your non-steroidal anti-inflammatory drugs. If you've never heard of NSAID, that's what that means. So COX-1 and COX-2 make prostaglandin and thromboxanes. What enzyme makes leukotrienes? I don't know why students missed on this on the exam last term. What enzyme makes leukotrienes? COX or LOX? LOX. So LOX enzymes make leukotrienes. COX enzymes make, I'm never going to forget now. COX enzymes make the thromboxanes and the prostaglandins. All right, so this is from your textbook. I'm just going to skip this slide. It's showing the same thing, except in much more detail about how you make the prostaglandins and thromboxanes and leukotrienes. But here's showing aspirin. Aspirin actually is a competitive inhibitor of the enzymes. So it will actually compete for the active site. Oh, good. All right. I really want to get started on the next one.
Let's go home.
Dr. Farley, I'm gonna go.
All right, guys, study hard. Watch the cholesterol video before you come in tomorrow, maybe. Yeah, it's there.